Hi guys, this video is all about cell communication and the different ways that your cells send and receive signals around your body, and also the ways that cells of other organisms are able to send and receive signals and also amplify those signals so that just one small signal can have a really big effect in the cell and also in your body. So, um, first of all, this cell communication can be either um, stimulatory, which means it's going to like turn things on and make things happen, or it can be inhibitory, which means it's going to turn things off and stop them from happening. Both of them are equally important because you want to be able to activate things at the right times, and also you don't want them to be activated when they don't need to be activated, whether that's like the expression of genes or the production of proteins or anything else. You really just want to be as efficient as possible. So you have signals that will turn things on and signals that will turn things off. And then these signals could be received from other cells. They could be received from other organisms, or they could be received from the external environment. Every single organism is able to do this to respond to um, like chemical signals or environmental stimuli, even single celled organisms like bacteria. We're going to watch in class this video right here, about how bacteria are able to actually communicate with each other, but they can do something called quorum sensing, which is right here. Basically, they're able to sense their population size through cell signaling. And they can use that to actually trigger certain genes or certain traits to be expressed that are only really beneficial when there's a certain population size. One example is bioluminescence. So there are some bioluminescent bacteria that are really cool, but they will only like trigger that gene when they know through like signaling between bacteria that there are other bacteria around and that the bioluminescence is gonna like have an effect. So it's amazing that they're able to actually like know that through quorum sensing. So that's kind of what this was talking about here, with bacteria being able to coordinate activities that benefit large numbers of cells. Um, Other examples of this are the biofilm that forms on your teeth at night. So when you wake up after you watch this video, you might notice, maybe for the first time, that there's a like film that's on your teeth, which is why you should always brush your teeth in the morning. But it is actually a film of bacteria that have grown there overnight and replicated, because they replicate super fast. But through quorum sensing, bacteria are able to communicate with each other, that they grow in a single layer instead of actually growing like on top of each other. Um, and they're able to monitor like the density of the population that way, which is amazing. Okay, so for multicellular organisms, this gets to be, of course, a little bit more complex. Um, Within multicellular organisms, your cells are able to communicate with each other to support the function of you as a whole organism. Um, a really clear example of that is the endocrine system, which I'll talk about at the end of this slide here, but also it's happening within individual cells as well. One really cool example is epinephrine, which is adrenaline. So I'm going to highlight this. Um, what adrenaline actually does is stimulates the breakdown of glycogen in your cells. For a little review, glycogen is the sugar that is animal starch. So it's basically your stored up sugars that you didn't need at the time. So when it's broken down, it's broken into glucose. When it's broken into glucose, it can be used for cellular respiration. And when it does cell respiration, we just learned this, it makes ATP, tons of ATP super fast. And you need that if adrenaline is being floating around in your blood, because that's what happens when you're in like a fight or flight sort of situation where it's like you have to make it happen right now. This is when like incredible, like superhuman feats of strength happen, which like moms lifting cars off of babies and like crazy stuff like that. And you would have to have tons of ATP to do that. So epinephrine or adrenaline actually does that or allows for that by breaking down the glycogen that's stored in your liver. Just another thing about epinephrine, sorry. If you watch doctor shows like Grey's Anatomy or anything like that, they'll say like push another epi. That's an epinephrine shot to trigger like sort of a fight or flight sort of response in a patient who is um, maybe coding to get the cells to start to respond. Just interesting fact if you watch shows like that. Um, and we'll talk more about how this pathway works to activate glycogen breakdown in the next few slides. Cells can also communicate by being in contact with one another. So they can literally like pass things through their cell membranes. One cool example of this is plasmodesmata in plant cells, which are literally gaps in the cell membrane. I'll show a picture in just a second. But they're holes so that things can pass through because plant cells are right up against each other and they are surrounded by this super rigid cell wall. So they have a space for materials to actually pass through from cell to cell. Like they're right up like this. And then also, multicellular organisms have this amazing skill to do long distance signaling throughout their body, which we use our blood to handle that, and our endocrine system. So, hormones are just chemical signals that travel through your blood so that a chemical signal that was produced in your brain can eventually have impacts on your thyroid, your pancreas, your reproductive organs, all over your body. So we'll talk more about the endocrine system after Thanksgiving break, but it is a really cool example of how cell signaling works at a larger scale instead of the cellular level. All right. So here's some pictures that are related to that. There's the first one is just showing like cells being right up against one another and the gaps between cells that allow signal the molecules to pass. So in animal cells, this is called a gap junction. And you can see they're like literally the little gaps. In a plant cell, it's a little bigger because it's a gap in the cell wall also. So this is called plasmodesmata. But either way, they have the same function, which is just allowing materials to pass literally directly into the adjoining cell. For this one below, this is showing cell to cell recognition. So you have cells that have specific glycoproteins. So these right here are glycoproteins. Usually, they could be like a protein complex, but typically they're glycoproteins, which are just membrane proteins that have sugar sticking off of them. And then you have other cells that have receptors that match with those specific glycoproteins that then send signals to that cell once they connect to be like, oh, this needs to happen. A really good example of this sort of cell to cell recognition happens in your immune system um, because it's how your cells not only recognize foreign invaders, but also how your cells recognize each other. So you have a complex of proteins on all of your cells that are you, that your body recognizes as you. It's like, oh, that's the right complex. That's me. That's supposed to be here. If cells don't have that like complex that allows your body to recognize your cells as you, then the immune system will start to trigger an immune response and you'll feel sick. This is why you can't get blood transfusions that are a different blood type than you, because your blood type is a specific type of glycoprotein. So if you're A, you have A glycoproteins. If you're B, you have B glycoproteins. If you're AB, you have A and B glycoproteins. And if your white blood cells start to recognize the wrong glycoproteins, it will trigger an immune response, and that will could even like send you into shock. So it can be really serious. But the good news is that your body's really good at recognizing what is you and what is not you, and that keeps you safe most of the time. So there you go. Okay, let's keep going. Here's a diagram that shows more signaling examples, especially in multicellular organisms. So you have a few different things happening here. The first one, A, is showing paracrine signaling. Paracrine just means that it is going to affect cells that are close by, which is why it says local signaling at the top. I'm just going to make a note of that this is affecting nearby cells. And then synaptic signaling is only related to the nervous system. This is nervous system. And then endocrine system is, it means that it has to use the bloodstream or the circulatory system in order to get to its destination. So it's going to be a much longer distance signal. So I'll say this uses bloodstream to send signals. So you have just these examples here in the images. Synaptic signaling is pretty cool because it's related to neurotransmitters, which in a lot of ways is related to how you feel and different emotions. You have things like serotonin, dopamine that make you feel nice, melatonin, which makes you feel sleepy. And they happen right in a tiny little synaptic gap in between neurons. They're produced by a neuron, stored in little like vesicles, released when they're signaled to be released. And then the target cell, which is another neuron, a sensory neuron, will receive that signal and be like, oh, time to trigger like the wind down or time to trigger feelings of happiness. They basically make you feel things. Lots of different endocrine hormones exist. You have like estrogen, testosterone, but also things like thyroid hormone, which regulate your metabolism. And then there are other hormones that are called tropic hormones, which their job is just to trigger the release of other hormones. So there's many different levels to this. It's a very complicated body system, but it's a really important one for making sure that you function normally. And if you've ever had any sort of like issues with hormones, you know that they can have really, really big effects on your entire body, really overarching things. So, okay, let's move on. Alrighty, so we're going to go back down to the cellular level now, and we're going to talk about when a signal is received, what happens. And there are 3 steps to that. The first step is reception, which is just the receiving of the signal. Typically, most of the signals that we're going to talk about do not actually enter the cell. Some of them do, but for most examples from here on out, we're going to talk about cells that bind to a receptor, which is a membrane protein. So, kind of just like enzymes worked, they have to be the right complementary shape for the receptor. We call the signal molecule a ligand, and that'll be on a future slide, but it's the word for that. The second step is transduction, and this is the longest step. So What happens is after the signal binds, that causes a whole bunch of changes that happen one step at a time in a cascade throughout the cell. So typically, the receptor, once it binds to a ligand, will change shape, and that shape change will trigger activation of other signal molecules that will trigger more signal molecules and more signal molecules and more signal molecules until eventually you get to the end of the chain, which will result in the response, which is step 3 here. Before I go off of transduction, I do want to say that often these signals are amplified, which I'll talk more about in the next few minutes. But a lot of times there can just be one signal received, but the cell is going to end up doing a lot of things because the signal is amplified by this cascade. And there will be a diagram that will kind of help with that. Finally, the last step is response. So usually what that means is that the message reaches the nucleus or the part of the cell that needs to start to be activated. A lot of times it is the nucleus because it's going to change the expression of a gene, either like starting to express the gene or stopping expressing the gene. So the example here is that when you eat lactose, um, your body needs lactase to break it down. So there will be a signal cascade that will trigger the nucleus to turn on the gene for lactase and start to do the processes of transcription and translation, which we haven't talked about a ton yet this year, but you might remember from freshman bio that they produce proteins from genes, transcription and translation. So then the response will be, okay, we're producing lactase now, or lactase in response to the signal. Okay, and now we're going to dive in, of course, to each of these 3 things separately. Before we do, though, here is an example image for those of you who are more visual of how this works. So the signaling molecule here is the ligand. Notice how it's the perfect shape for the receptor molecule. The receptor molecule is embedded in the cell membrane. And then what happens is that the signal never actually enters the cell, but because it binds to the receptor, it causes changes in the receptor and also in the future molecules that lead to eventually a cellular response happening. And I'm going to just make a note here that a lot of times, often response is transcription of a specific gene so that a specific protein is produced to do a specific job. Okay. Now, let's talk about receptors. So, like I've already said, the receptor and the signaling molecule, which is the ligand, and that's finally written right here, that's the signal molecule, have to be complementary, just like an enzyme and a substrate. They have to fit perfectly together. Usually, receptors are plasma membrane proteins most of the time. The ligand, when it binds, is going to usually trigger a change in the shape of the protein receptor, which then like allows it to interact and change the shape of future proteins that will eventually like carry the message, almost like telephone, playing telephone, except for the message like is right. They don't mess it up like we do when we play telephone. But the shape changes eventually will trigger the response as the cascade passes on. Sometimes there are extra molecules worked in there. One example is a G protein. So there's a specific type of receptor called a G protein coupled receptor. And what this is you have the receptor that binds to the ligand. And there's also a like separate protein that's called a G protein. The G protein needs to be activated by a molecule called GTP, which is super similar to ATP. It is just the energy source that this specific protein uses. The G protein is on the inside of the cell membrane and just sort of slides along. This will be, I'm actually going to show the picture now. Here's the picture of this. So you have the G protein coupled receptor, which is the purple molecule, and then the G protein is the dark green oval molecule. See how the G protein's on the inside of the cell membrane. When the receptor is activated because the ligand binds to the receptor, that activates the G protein. So it basically just switches out its GDP, which is just like ADP, the inactivated form, with GTP, which is the energy rich form. Then what the G protein can do is sort of act as a messenger to start the cascade. So it will go over to an enzyme, activate that enzyme, and then the enzyme that was activated by the G protein can start this whole like cellular response cascade. It doesn't always have to be a whole cascade. It could be short, but it's often a cascade. And then once that GTP is used to activate the enzyme, it's converted back to GDP, the inactive form, and the whole process can start back over again. And that's a G protein. Let me go back up here and finish this. I mentioned this briefly, but most receptors are found in the cell membrane and not inside the cell, but sometimes some signaling molecules actually do enter the cell. And those are things typically that can pass through the cell membrane, like lipid hormones, which steroids are an example. And then also thyroid hormones do this too. So things like testosterone and estrogen actually can pass through the cell membrane because they're fats. So it's a lot easier for them to get into the membrane. And then once they're inside the cell, the testosterone and the estrogen, they bind to receptors inside the cell. Not every cell has receptors for these hormones. So if they don't have receptors, then they just don't respond. And the ones that do have receptors do respond. I didn't mention this, but I just want to say it that it's on here that the G proteins, just going back up here, are used often by yeast. They're also used in the epinephrine adrenaline pathway, and they're used by neurotransmitters. So we're going to do like kind of a deep dive into the epinephrine pathway in just a minute. You can see how it works. And then bacteria that cause things like botulism and whooping cough, which are really yucky bacteria, they actually work by interfering with the function of G coupled protein receptors. And that's part of why they're such bad news, because the cells then are not able to respond to signals, even though the signal is received, which is not great. All right. Talked about G proteins. Okay. So now let's talk about how this transduction process actually happens. And typically, how it works is through phosphorylation and dephosphorylation using ATP. So protein kinases are enzymes whose job it is to add phosphates to things and therefore activate them. So I'm just going to make a note over here that says kinases add phosphates and activate proteins. And the phosphates come from ATP. And that in turn causes activation, which typically is then used to activate another one down the chain. Also, there are enzymes that are responsible for turning off the pathway or turning off proteins, and those are called phosphatases. I'll label them over here. I'm going to say that phosphatases remove phosphates, which means that they deactivate proteins. So these guys, the kinases and the phosphatases, are really responsible for regulating a ton of the transduction process because they're the main things that turn on and off the enzymes that are the steps in the pathway. There are also molecules called second messengers, and they're super cool because their job is to be triggered to amplify the signal, to spread it out so that there can be many different cascades happening at the same time in response to just one signal. They are not proteins. They're typically like really small little proteins. I'm sorry, not proteins. They are not proteins. They're typically really small molecules or even ions that can be triggered and then easily spread throughout the cell to like amplify and make the signal even bigger so that the response can be even bigger. Two examples of these are cyclic AMP, which we just write as CAMP, which is found in liver cells, and its job is to help break up glycogen. So you might be able to guess that it's part of the epinephrine adrenaline pathway. And then calcium ions are really important in muscle cell contractions and also in secretion of different materials and in cell division. But a lot of times we think about muscle contractions as being like the best example of when calcium is needed to help amplify that signal. And think about how fast you need to contract your muscles if you're exercising super fast. So the only way that is able to happen is using second messengers to make the signal of contract, contract, contract way bigger so that the cells can do it fast enough. All right, let's look at some pathways. So here is just a example pathway. It's not a specific signaling molecule, but it just kind of shows you what's happening. So you have these different kinases, which remember the kinases are responsible for adding phosphates and thereby activating the next molecule. So see in the pathway how the kinases are actually what they're phosphorylating or activating is the next one in the chain. And they're using ATP to get that phosphate. So they're taking it off of ATP and putting it onto the kinase. And then ADP is what's left behind. This whole process is called a phosphorylation cascade because what it's doing is it's adding phosphates to activate each kinase. And at the end, what you have is an active, a whole bunch of active proteins, and then a cellular response that is specific to what the signal was. The PPs down in the bottom, the little gray ovals, those are the phosphatases. So this is a phosphatase, which is deactivating kinases to restart the pathway. Because if you have them active all the time, the pathway is not going to be able to like restart and reactivate to get another signal. So you need those phosphatases to come behind and deactivate the kinases so that the whole process can start over again next time a signal comes. All right. Let's look at this one. Okay, so here's an example that shows the adrenaline pathway, and it also shows an example with a G protein. So when epinephrine binds to the receptor, that causes the G protein to become activated. That's why it has the GTP on there. That means it's activated. It only is activated when the signaling molecule binds. The G protein is then used to activate this enzyme that's called adenyl cyclase. And then adenyl cyclase uses ATP to activate cyclic AMP, which is this is the second messenger. So this job is to activate, I'm sorry, not activate, amplify the signal. And there wouldn't be just one cyclic AMP molecule. There would be many cyclic AMP molecules that all were becoming activated by adenyl cyclase. So that then when cyclic AMP goes to activate protein kinase A, it's actually many cyclic AMPs activating many protein kinase A's, so that this process, this cascade, is happening in many different cascades in the same cell at the same time to make the response bigger and faster. And then eventually you have this picture doesn't show the whole like kinase cascade, but it is there. And eventually that leads to a response, which is the breakdown of glycogen in liver cells. So now finally, we've arrived at the response after all the transduction. Typically, the response is triggered in the nucleus, but it could also be triggered in the cytoplasm. So that would be regulating the activities of the cell not related to like coding for a specific gene. I'm just going to make a note here that transcription is converting DNA to mRNA to code for specific traits or proteins. Um, the response can be fine-tuned in a few different ways. So one of them is the amplification, where each step produces more products than there were signals. And that's how the adrenaline pathway works. Also, what's cool about this is that it's specific a lot of times to cell types. So for epinephrine or adrenaline, it has one effect on the heart and a different effect on the liver, because those two cell types have different proteins that are going to respond to epinephrine. So even though it's the same signal, the different cells, depending on what protein receptors and links in the chain they have, can lead to really different responses. So in the liver, like we've talked about, it's going to cause glycogen breakdown into sugar so that that sugar can be used to make energy. Whereas in the heart, it's going to lead to muscle contraction to pump more oxygen to the cells so that they can produce more energy by doing aerobic respiration using that oxygen. So it's really cool that the response can be fine tuned by changing which receptor molecules and which like members of the cascade, which kinases in the different cells of your body. So here's an example of how amplification works with epinephrine. So at the beginning, you have just one molecule of epinephrine binding to one G protein coupled receptor. That's literally one molecule. By the end of the cascade, you can see that every time it's getting bigger and bigger and bigger. And here's CAMP, cyclic AMP. This is the second messenger. It's called a second messenger because the epinephrine was the first messenger. So this is kind of like the second piece that like restarts the process, or maybe not even restarts it, but like triggers it to be even bigger. So you can see by the time you get to the end of the cascade, you're looking at a response of 10 to the 8th power molecules where you started with only one, which is crazy that your cells are able to amplify the signal that much to produce so many things from just one original signal. And that's why a molecule like epinephrine causes such a huge response in bodies, because the signal is amplified so much so that so much can happen in response to just the one molecule. All right, last thing is that there are a lot of issues that can occur with signal transduction going wrong. So if the transduction pathways are blocked or defective, there can be a whole bunch of different diseases that can result depending on what cell type is having an issue, what pathway is having an issue. Just a few examples are listed here. Of course, cancer is a group of diseases. It's not one disease, so it will have um, different or different cancers are going to be caused by different issues. So you can't just say like, oh, all cancer is caused by issues with signal transduction, because it's not the case. Also, certain drugs and toxins and poisons and pesticides are able to alter signal transduction by interfering with the receptors usually. One like kind of cool example is antihistamines, which are like anti-itch usually creams. They block the pathway, sort of like a competitive inhibitor to the receptor, so that the signal pathway doesn't even actually start. That you don't feel those things because your cells aren't getting the signals because they're being blocked by the antihistamine or the anesthetic, which is amazing. Okay, so that is the story of signal transduction and how your cells are able to get little tiny signals and make them into big, huge responses, not only in the cell, but also in your entire body, which is an amazing thing. So thanks for watching, and I'll see you soon.